Alright, so continuing our topic of discussion, that is the circulatory system, we are going to take a look at the blood groups or the blood grouping system. And the most used blood grouping system that we know is the ABO blood grouping system. Now, when we talk about ABO blood grouping system, the basis of the ABO blood grouping system is the presence or absence of two antigenic determinants on the surface of the red blood cells or the erythrocytes. Now, these antigenic determinants can either be A antigen or B antigen, right? So, if the A antigen is present on the surface of the RBC, we call that person to be the type A. If a B antigen is present, then the person is of type B. If both of them are present, then we call that person to be type of AB. And if neither A nor B is present, then that person is of the type O blood group. Now, what are these antigenic determinants? Let's look into that. So, these determinants on the surface, they are nothing but sugars and sugar derivatives. So, if we take a look at the O-type blood group, then you can see it has these combinations of different sugars. And if you look over here, then we have one fucose molecule, two galactose molecule, and one N-acetyl glucosamine, which is a sugar derivative. So these are the molecules that combine to form the antigenic determinant on the O blood group, right? When it is A, you have all of these, but you have an extra, which is the N-acetyl galactosamine. And in B, you have 3 galactose instead of 2 galactose. And when you have both the A and the B together, that is, you have the A and the B, then you have the AB blood group, that is, having both the determinants of A and the determinants of B. Now, holding on to that thought, another very important aspect is the presence of antibodies related to the blood group that we are talking about. Now, when the blood group is of A type, the antibodies present in the plasma would be anti-B. Now, if you think logically, then of course, if anti-A is present in a person who is of A type, of the blood group A type, then those antibody that are present, let's say this is the anti-A, and the person is having A antigen on its surface. If this was the case, then this antibody would go and bind to the blood cells, and that is going to lead to the destruction of the RBC. That is why you cannot have anti-A in a person who has the A determinant on the RBC. Now, similarly, when the blood group is B, when the red blood cell is having B type and B-type determinant on the RBC, then you cannot have the anti-B. You need to have the anti-A. Again, the same thing. Anti-B is going to, interact with the B determinant and going to destroy the RBC. But what happens when you have both A and B? When you have both A and B, in that case, you cannot have either anti-A or anti-B antibodies. So that is why none is present. And finally, when you have the O antigen or the O blood type, then you can have both anti-A and anti-B, because neither determinant A or determinant B is present, right? So these are the few things you need to keep in mind. Along with this, another two very important thing that you can that you need to keep in mind is the blood type of O. The blood type of O is referred to as the universal donor. That is, it can donate blood. Any person with O blood group can donate blood to all the other blood types. Whereas AB is referred to as the universal acceptor or universal recipient. It can accept blood from any blood type because it is not having any antibodies. Now, you can have a question that in the person who is having the O blood type, he or she would have anti-A and anti-B altogether. So how can the O blood type person give blood to A or to B? This question can arise. Now, when we are talking about donation, it is either RBC donation or plasma donation. So according to this, the blood groups are selected. Right. So next, what we are going to see, we are going to see another antigenic determinant, which is called the RH factor, and we are going to take over from that. So all of that in the next video.